Welcome to Switzerland. Did you know that our country is not just a great place to visit, but also your first-class business location? We are centrally located in Europe and we have the world's best infrastructure. Oh, and the train is always on time, so let's take a journey. Did you know that Switzerland has 4 official languages? Ja? No? Forse? Decidia? Don't worry, we also have excellent English skills. However, we don't spend all day chatting. We love to work. In 2012, we could have voted for longer holidays, and we voted against them. It's crazy, isn't it? But we're not crazy. Our economy and government are among the world's most stable. That, combined with our outstanding talent pool and a deregulated labor market, makes Switzerland the most innovative and competitive nation in the world. Our Swiss dual education system provides an excellent mix of both vocational education and apprenticeships. Now let's take a look at industry. Maybe Germany, the United States, and China are well known for their industrial output. But did you know that Swiss industrial production per capita beats all of them? Sorry, guys. Our country is small, but hey, 3 Swiss companies are among the global top 20 most valuable companies. Furthermore, we offer the best quality of life. That's why 3 Swiss cities are among the world's best for the highest quality of living. In addition, Switzerland is one of the happiest nations on the globe. Perhaps because we eat on average 12 kilograms of chocolate per year? Switzerland is the best location for innovation, has stable political, economic, and financial conditions, has the highest living standard, and welcomes your business. So tell us now, when will you invest in Switzerland? Switzerland Global Enterprise, enabling new business. Find out more at s-ge.com.
For us in the Aditya Birla Group, Odisha has been one of our most preferred investment destinations. Chief Minister Shri Naveen Patnaik's exceptional leadership has been the singular factor in the rapid progress of Odisha. The state government has leveraged its competitive advantages, including its progressive policies and governance framework, are a big pull factor.
Welcome you to Odisha. Come, join the juggernaut.
In healthcare, a vast ecosystem is helping therapy innovators become more patient-centric and more effective than ever before. Cytiva supports researchers, biopharma, and clinicians in the pursuit of more targeted treatments. In discovery, speed and accuracy are everything. The faster and smarter researchers can work, the more patients benefit. To ensure that new, more targeted therapies become reality, we're equipping biomanufacturers with the tools and expertise they need to produce cost-effectively and quickly. How can tens of thousands of individualized patient doses be manufactured quickly, efficiently, and securely? The answer, with a healthy dose of expertise and innovation. We're creating processes and solutions that are scalable and address complex needs. We work to deliver diagnostic tools and other technologies to bring the right therapy to the right patient at the right time, so that life-saving medicines come within the reach of more people and could save more lives. Cell culture, protein purification, biomarker imaging, and analysis. It may not be what most think about when envisioning the future of healthcare, but this is what we do. Our part in changing the world. Advancing and accelerating. Discovery, manufacturing, diagnostics, future therapeutics.
road to regulatory compliance is long and filled with choices. With a combination of standards, process, and service, USP can help guide your path to compliant, quality products. Trust the standard that sets the benchmark for medicines. Our robust and collaborative scientific process is part of our comprehensive approach. We offer reference standards that are tied to USP monographs to help you minimize risk and enhance your confidence in compliance. Reduce time and resources spent developing in-house standards and streamline your path to regulatory compliance. We work with independent, volunteer scientific experts who rigorously review and approve our standards, which undergo testing by USP and other laboratories around the world. Ongoing testing of our standards helps ensure the quality of your product over time, and only USP reference standards are linked to official USP NF monographs that provide specifications for the identity, purity, and potency to meet FDA requirements without further in-house qualification and the needs of our global customers. With USP, you have access to our in-house scientific experts to guide you every step of the way. In addition to our best-in-class USP reference standards, we offer training, education, and other services. Our online resources help smooth your path to regulatory compliance. Our standards provide precise testing and validation guidelines, as well as reference samples for testing. Drugs can be made consistently every time. We are more than reference standards. We provide a unique combination of standards, process, and service. From buying to applying standards, we support your bottom line with reference standards that have been rated best in class by our customers. Talk with us to find out how we can help you navigate your journey to regulatory compliance. Contact USP at usp.org.
Welcome to Switzerland. Did you know that our country is not just a great place to visit, but also your first-class business location? We are centrally located in Europe, and we have the world's best infrastructure. Oh, and the train is always on time, so let's take a journey. Did you know that Switzerland has 4 official languages? Ja? No? Forse? Decidia? Don't worry, we also have excellent English skills. However, we don't spend all day chatting. We love to work. In 2012, we could have voted for longer holidays, and we voted against them. It's crazy, isn't it? But we're not crazy. Our economy and government are among the world's most stable. That, combined with our outstanding talent pool and a deregulated labor market, makes Switzerland the most innovative and competitive nation in the world. Our Swiss dual education system provides an excellent mix of both vocational education and apprenticeships. Now let's take a look at industry. Maybe Germany, the United States, and China are well known for their industrial output. But did you know that Swiss industrial production per capita beats all of them? Sorry, guys. Our country is small, but hey, 3 Swiss companies are among the global top 20 most valuable companies. Furthermore, we offer the best quality of life. That's why 3 Swiss cities are among the world's best for the highest quality of living. In addition, Switzerland is one of the happiest nations on the globe. Perhaps because we eat on average 12 kilograms of chocolate per year? Switzerland is the best location for innovation, has stable political, economic and financial conditions, has the highest living standard, and welcomes your business. So tell us now, when will you invest in Switzerland? Switzerland Global Enterprise, enabling new business. Find out more at s-ge.com.
For us in the Aditya Birla Group, Odisha has been one of our most preferred investment destinations. Chief Minister Shri Naveen Patnaik's exceptional leadership has been the singular factor in the rapid progress of Odisha. The state government has leveraged its competitive advantages, including its progressive policies and governance framework, are a big pull factor.
I welcome you to Odisha. Come, join the Juggernaut.
In healthcare, a vast ecosystem is helping therapy innovators become more patient-centric and more effective than ever before. Cytiva supports researchers, biopharma, and clinicians in the pursuit of more targeted treatments. In discovery, speed and accuracy are everything. The faster and smarter researchers can work, the more patients benefit. To ensure that new, more targeted therapies become reality, we're equipping biomanufacturers with the tools and expertise they need to produce cost-effectively and quickly. How can tens of thousands of individualized patient doses be manufactured quickly, efficiently, and securely? The answer, with a healthy dose of expertise and innovation. We're creating processes and solutions that are scalable and address complex needs. We work to deliver diagnostic tools and other technologies to bring the right therapy to the right patient at the right time, so that life-saving medicines come within the reach of more people and could save more lives. Cell culture, protein purification, biomarker imaging, and analysis. It may not be what most think about when envisioning the future of healthcare, but this is what we do. Our part in changing the world. Advancing and accelerating. Discovery, manufacturing, diagnostics, future therapeutics.
To regulatory compliance is long and filled with choices. With a combination of standards, process, and service, USP can help guide your path to compliant, quality products. Trust the standard that sets the benchmark for medicines. Our robust and collaborative scientific process is part of our comprehensive approach. We offer reference standards that are tied to USP monographs to help you minimize risk and enhance your confidence in compliance. Reduce time and resources spent developing in-house standards and streamline your path to regulatory compliance. We work with independent, volunteer scientific experts who rigorously review and approve our standards, which undergo testing by USP and other laboratories around the world. Ongoing testing of our standards helps ensure the quality of your product over time, and only USP reference standards are linked to official USP NF monographs that provide specifications for the identity, purity, and potency to meet FDA requirements without further in-house qualification and the needs of our global customers. With USP, you have access to our in-house scientific experts to guide you every step of the way. In addition to our best-in-class USP reference standards, we offer training, education, and other services. Our online resources help smooth your path to regulatory compliance. Our standards provide precise testing and validation guidelines, as well as reference samples for testing. Drugs can be made consistently every time. We are more than reference standards. We provide a unique combination of standards, process, and service. From buying to applying standards, we support your bottom line with reference standards that have been rated best in class by our customers. Talk with us to find out how we can help you navigate your journey to regulatory compliance. Contact USP at usp.org.
Welcome to Switzerland. Did you know that our country is not just a great place to visit, but also your first-class business location? We are centrally located in Europe, and we have the world's best infrastructure. Oh, and the train is always on time, so let's take a journey. Did you know that Switzerland has 4 official languages? Ja? No? Forse? Decidia? Don't worry, we also have excellent English skills. However, we don't spend all day chatting. We love to work. In 2012, we could have voted for longer holidays, and we voted against them. It's crazy, isn't it? But we're not crazy. Our economy and government are among the world's most stable. That, combined with our outstanding talent pool and a deregulated labor market, makes Switzerland the most innovative and competitive nation in the world. Our Swiss dual education system provides an excellent mix of both vocational education and apprenticeships. Now let's take a look at industry. Maybe Germany, the United States, and China are well known for their industrial output. But did you know that Swiss industrial production per capita beats all of them? Sorry, guys. Our country is small, but hey, 3 Swiss companies are among the global top 20 most valuable companies. Furthermore, we offer the best quality of life. That's why 3 Swiss cities are among the world's best for the highest quality of living. In addition, Switzerland is one of the happiest nations on the globe. Perhaps because we eat on average 12 kilograms of chocolate per year? Switzerland is the best location for innovation, has stable political, economic, and financial conditions, has the highest living standard, and welcomes your business. So tell us now, when will you invest in Switzerland? Switzerland Global Enterprise, enabling new business. Find out more at s-ge.com.
Yeah, good evening, Dr. Bhavani.
Sir, namaskaram, sir. Modi sir
Namaskar,
and
namaskar,
Dr.
namaskar.
Okay, sir. How are you, sir?
Yeah, fine, fine.
Yes. Sir, namaskaram, sir. Okay, sir.
A very good evening to all, and welcome to the session on blue economy. As we know, the sea is undoubtedly the treasure of assets and benefits for humankind. As the world population is increasing, so are the demands and the needs. There may be a time when land alone will not be able to provide us optimal satisfaction. Hence, turning to the ocean is surely indispensable. Oceans could be a crucial importance in ensuring food security, health security, and energy security. And India is being vastly surrounded by this resource. We have the Bay of Bengal in the east, the Arabian Sea in the west, the Indian Ocean to the south, and therefore there is an immense option of exploring this resource. Blue economy, or marine biotechnology as we know it, is focused on research and development of technological applications of living marine organisms, their derivatives.
I'm waiting for them to ask me to talk.
But on the other hand, it also encompasses the protection that is needed for the marine environment for future generations. We at TBT and BioRac are aware of this and recognize the enormous potential that the development of blue economy holds for our country. Part of blue economy initiatives, the Department of Biotechnology has developed consortium programs, virtual centers with strong linkages proposed. There have been a number of discussion dialogues with different companies and academic institutes which focus on product technology development in the area of aquaculture and marine biotechnology. Initiatives have been taken to establish the state of art for marine biology and biotechnology for exploring ocean resources towards the efforts for harvesting the blue economy. And today, we are very fortunate to have a panel of eminent experts who will cover different aspects of exploring the marine economy, what options do we have, and how can this be made a sustainable economic growth potential for the country. With this, I would like to invite our first speaker for the evening, Dr. Yogesh Shoche from National Center for Cell Sciences, Pune, to deliver his talk. Sir, I will request you to kindly share your screen and start with your presentation. Thank you very much. Dr. Shoche, are you there?
Hello.
Hello, good evening, Dr. Shoche. Can you please share your screen?
Yeah, sure. 1 second.
Yes.
Can you hear me?
Yes, loud and clear. Dr. Shoche, we still can't see your screen. Request you to kindly share the screen and your presentation.
Yeah, in case if you have any problem, then we can share from our end.
Dr. Shoche, if you're having any issues, should we play it at our end? Yes, we can see your screen, I think. Can we go to the full screen mode?
So do you have any issue, Dr. Sochi?
हेलो सर, नहीं मेरा मुझे कुछ सुनाई नहीं दे रहा है. कैन यू सी माय स्क्रीन नाउ?
Yes, sir. We can
Can
see
you
the
hear
screen
me?
now. Yes, we can hear you as well.
Okay, fine. Yeah, okay, yeah. There is some internet problem anyway. Yeah, so at the onset, I would like to thank the organizers for giving me this opportunity to make this presentation here. And what I'm going to talk about is exploration of marine microbial resources for blue economy and what DBT has done in the last couple of years on these lines. So as we all know, Earth is a blue planet with 70% water, and marine microbes contribute 50% global primary production. And in terms of microbes, it is home to polyextremophiles, that is thermophiles, halophiles, barophiles, and this is the gold mine for various novel compounds which include health applications, antibiotics and other drugs, then enzymes. The well-known example is thermostable polymerases, then biofuels and functional foods. So there is a tremendous scope for marine products, marine microbial products in blue economy. And India, if we talk about, it is blessed with 3 oceans, Indian Oceans, Arabian Sea and Bay of Bengal. It has more than 8,000 kilometers of coastline, and it has 2.02 million square kilometer of exclusive economic zone. So, and in this zone, there are diverse ecosystems like estuaries, coral reefs, marshes, mangroves, deep sea. Recently, people have also discovered some hydrothermal vents in the deep sea. So the opportunities are immense. And DBT in the last couple of years have taken many major initiatives for marine exploration. It started more than a decade ago, where DBT supported a project to explore microbial diversity, where expertises of NCCS and NIO were put together. This project was a preliminary project to understand and study the marine microbial diversity in the western coast of India in collaboration with NIO. And this project resulted in several publications describing bacterial and fungal diversity of the western coast. Plus, we also identified 3 new species of bacteria from estuaries, mangroves, and sediments of from the western coast. DBT has also established national facility for marine cyanobacteria at the Bharati Dasan University, And as we know, cyanobacteria are very important source for biofuels and many other products of biotechnological importance. So this facility is supported by DBT and is present at Bharatidasan University at Tiruchirappalli. Taking this further, DBT had taken up one major initiative in microbial exploration, where 9 institutes participated and generated 1,000 isolates per month, making it to over 200,000 isolates over the period of 3 years. And with the help of a participating company, for participating industry, these microbes were screened for various bioactive compounds for human health, that is anti-infective, anti-cancer, anti-diabetic, and anti-inflammation. And amongst other ecosystems, marine ecosystems was an important component of this exploration, where MSSRF contributed isolates from mangroves, and National Institute of Oceanography, Goa, contributed marine sedimental isolates. And total 40,000 isolates from these 2 ecosystems were screened in this exercise using HTS platform that was available with industry that time, out of which 1,500 showed potential therapeutic activity in any of the 4 areas listed. These were further analyzed and grown in large quantity, and attempts were made to purify the active component. And this exercise has then resulted into 6 pure compounds which are available right now under either of the 4 categories, and plus 4 postulated novel structures. And these all are from the marine sources, which include leads in the areas of anti-cancer, anti-infective, anti-inflammation, and anti-diabetic. So this is the outcome of this major initiative by DBT, which was taken in 2007. Then, taking clues from this, currently DBT has initiated another major program, which is Marine Bioresource and Biotechnology Network. NCCS is coordinating this program, and there are participating institutes. There are total 6 participating institutes, and these in turn are collaborating with other institutes, making a big network program, and where each institute is looking for different bioactivities, which include microbiome enzymes, which will be done by Goa University. They will use bioinformatics approaches for prospecting microbial enzymes, anti-cancer. Dr. Sajivan from Cochin Institute of Science and Technology would be leading this, and they have some specific screens for anti-cancer compound and dereplication platform, and they will be looking, with the help of CDRI, they will be looking for novel anti-cancer compounds. Then Goa University, Dr. Savita Kelkar, along with NCPR, they will be looking for pigments from marine microbes, which have again a very good commercial value. Then there is a component on looking for antifouling compounds, marine antifouling compounds, and this will be looked after by Satyabama Institute of Science and Technology, and IIT Mumbai would be involved in development of expression vectors for the production of marine products. And then there is also another component of looking for bioactive enzymes and other bioactive molecules, which will be looked after by CSM-CRI. So this is a big network project that has been initiated by DBT. In addition to anti-infective, this component would also look for other products of industrial importance from marine organisms. DBT has also proposed to start a DBT NCCS Marine Biotechnology Research Laboratory at Baitul, Goa. And though in a long term, many activities are planned to be taken up at this place, presently the immediate focus will be on drug discovery, biorepository, and genomics of marine organisms. So these are the activities that will be taken up at proposed DBT NCCS Marine Biotechnology Research Laboratory. Further understanding the importance of polar region, DBT has expanded the its a purview beyond oceans to polar region, North Pole as well as South Pole. And with the help of Ministry of Environmental Sciences, it has proposed a DBT MOS Center for Polar Sciences, and this center would look at diverse activities. It would look at cold active microbial enzymes. It would study the relationship between climate changes in the permafrost. It would also look for potential microbes, potential infectious agents coming out due to melting of permafrost, because there is a major global concern. So this center would look at that. It would also look at secondary metabolites, antibacterial, antimicrobial compounds. It will look at the horizontal transport of AMR genes. It will also look at ancient biomolecules in the sediments and ice core, and it will also look at the pathogen evolution. So these are the multiple activities which will be taken up by this DBT MOS Center on Polar Sciences that is going to be set up very soon. And so these are the various DBT initiatives that are proposed under the different marine biotechnology programs by DBT. And I would like to thank you for your patience. And once again, thanks organizers for allowing me to make this small presentation over here, and regret the initial technical difficulties. Thank you once again. Hello.
So, thank you very much, Dr. Social, for my presentation and providing very valuable information. Thank you. Now I request Dr. Ajay Parbar, ILS, to kindly Dr. Ajay Parbar, please.
Thank you, Dr. Manoj and Shilpi, for inviting me for this particular discussion point. I think Jogesh has already covered a lot of ground on various initiatives of the Department of Biotechnology in the marine and coastal coastal areas. And whatever you do, unless until you look into the people who are living in the coast or living elsewhere, I don't think that any technology intervention is going to help. So my focus on what are the science and technology options for ensuring a coastal area prosperity. And the globally also the major populated cities are in the coast which are extremely vulnerable, and it is densely populated. And if you look into the Indian context, the coastal ecosystem is a major ecosystem. Like India has 7,500 kilometers long coastline, including 2 islands. Then we have 200 nautical miles of exclusive economic zone. And this is a link between the terrestrial and aquatic ecosystem or the marine ecosystem. They have diverse ecosystem function, and that is why we are all talking about how do you take the advantage of the system for ensuring people's benefit. And this is what I mentioned, of millions of those poor families who are residing in this. But over the last few years, there has been significant level of destruction which has happened in the coastal area. The land use pattern has altered. I think something like about 280 million people live within the 50 kilometer of the coastline. That's a quite huge number of people. And something about 3,600 fishing villages or hamlets are available. and about 4,120 square kilometer of the mangrove. So from all point of view, coastal systems is a major ecosystem which need to be resourced. And so definitely, so this coastal, and I think as already Yogesh has mentioned, coastal and marine ecosystems, estuaries, coral reefs, marshes, lagoons, or mangrove forests, seagrass bed, these are potential biological resources which can be exploited for ensuring a better societal thing. But there are enormous of challenge in the coastal zone. Like rapid urbanization is creating a lot of depletion in the natural resources. There are competition, conflicts in the resource use, degradation, loss of flora and fauna, and there are whole lot of challenges which are happening in this domain. In this, I think Yogesh again has briefly talked about the mangroves, and mangroves essentially are what you call the lifeline of the coastal system. So they are a specialized ecosystem which is forms a link between the terrestrial aquatic environment, and these are diverse and productive ecosystem. This comprises of about 71 species spread over the 16 families. Although I think Indian coastline accounts for 8% of the world's mangrove area, but it comprises about 40% of the species which are reported globally. So in terms of species richness, the Indian mangroves are, but then there has been during last 25 years rapid degradation. Here, science and technology based interventions has provided tools and a lot of restoration has happened. If you see the satellite imagery within 10 years time, the Pichavaram mangrove forest in Tamil Nadu, which has been completely restored with almost about 3 times more mangrove coverage in that particular region. Then there are a few species which are available in the coastal areas which are of high potential. For example, there is a species called Salicornia brachyata. This species seeds has about 28% of the oil, 31% of the protein, and 5% of the fiber. And this can tolerate up to 70 gram per liter of the salt. So the question is that can we, like, given the type of salt salt concentration available in the coastal ecosystem, one these species we utilize for making some type of what is called the saline agriculture or grow the productive species in the salt affected soil. Similarly, there is a species like Artiflex, which is a huge, can be a fodder grass or something like that. So the question is that can you identify some of the species from this ecosystem which can become what you call potential source of productive agriculture system in the coastal region. Now coming to the high-end science or technology used, like for example, a work which was carried out at MS Swaminathan Research Foundation when I was associated there. This particular species grows, grows in almost over 3 times higher concentration of the salt water. So using biotechnological options, what we tried to look at is that we started looking at the genes which could be useful for developing transgenic plants or developing salt-resistant rice cultivars, and also which can be useful for the coastal area cultivation part. So we have used species like Abyssinia, Potracia, then these species and have isolated a large number of genes from this, so that now we have what you call the transgenic plants using the genes isolated from the mangroves. You would see the control plant and the two transgenic lines where this can be both salt and drought resistance. And these has also been, we have also started looking into some of the, like the nutritional deficiency is a major problem. So we also started looking at some of the iron or ferritin gene from the mangrove species, and we have developed transgenic rice using a endosperm specific promoter, so that even if you polish the rice, still the rice, the polished rice has 3 times more iron compared to the control plant. Similarly, the transgenic rice which we have obtained from the mangrove species, they have been transformed, they have been progressed into local varieties like white pony, higher 20, ADT 43, and higher 60. For what I would like to say is that once we think that these mangroves are absolutely not edible plant, but they are potential sources for genetic material which can be used for developing transgenic rice plant. And this rice plant has a potential of being cultivated in almost over 20 million hectares of land in the coastal areas of India. So this has a huge potential where science and technology options can be utilized for looking into the productive use of those resources for the betterment of the coastal area and other things. I think Yogesh has already briefed about your microbial bioprospecting, and there are enormous opportunity arises here. You can identify anti-cancerous activity and all these things. Like one of my colleagues, as Yogesh was saying in the network program, one of my colleagues was looking into the microbes from the hot springs of Odisha as well as the coral reef systems of Andaman Nicobar Island, and what you could identify some of the bacteria which has looked into biomolecules, and one of the products which are in pipeline for developing what we call wholesale livestock vaccine for whooping cough or respiratory disorder. And this has already been patented, and we are in discussion with one of the companies to make it to the next stage of commercialization. So as I said, that this is just one example of many what my colleagues has done. So they have identified. So this can be exploited for developing products. So there is Oceans has tremendous potential to take it up. Similarly, there is another resource which I think many people know, that the seaweeds is another useful resource which are available in the coastal areas. And now recently the government of India has launched what we call the National Seaweed Mission, and they are now going to set up about the government of India is going to set up about 6 seaweed parks where you can look into a whole range of products, starting from the food, or you can talk about the biofertilizers, biogas, bioethanol, so biodiesel, and I mean a whole lot of opportunities exist in that. And this could be a revolution from the blue revolution to what we call the seaweed could play a major role in making some of the product, which will also in turn bring in the economic prosperity of this region. So the major reason what I would say that in the recent initiative, as Yogesh has said, Department of Biotechnology is launching a major program on the coastal bioresources. Institute of Life Science in partnership with other institutes. What we thought is that unless and until you take the location specific issues and try to find out what are the type of potential resources they have. For example, we have selected 3 major ecosystems in Orissa. One is the Chilka Lake. All of you know that that is one of the largest saltwater lake in the world or in Asia. Then you have the largest mangrove vegetation called Vitarkanika mangroves. And the third location, which is that olive rudder turtles, which is another habitat. So we are going to look into the prospecting novel organisms or the organisms from those for potential use in various for either therapeutical or agricultural or some other use. So this project has already started. Our aim is to look into the both lower plants as well as the microbes from these 3 distinct habitats, and these 3 distinct habitats has characteristic features to develop some products which can be useful for the nation or even for the world. So looking at this forward, that as I said that with 7,500 kilometer coastline and also millions of resource poor people, and coastal region both has a challenge and has an enormous opportunity. Our coastal ecosystem are crucial reservoir of our biodiversity, but it all depends on us how to use technology applications for and in appropriate location specific manner for enhancing the lives and livelihood. And this I'm sure will help translating the bioresources to a bioeconomic culture. Thank you for this opportunity. I thank all of you.
Thank you, sir. That was really enlightening. And I think science and technology holds a great potential for harnessing the resources and promoting the marine economy and also translating the bioresources to bioeconomy, as you mentioned. Now, to hear about the biotechnological prospects that might be there from marine natural compounds, I would like to request Dr. G. Dharani from National Institute of Ocean Technology to kindly deliver his talk. Over to you, sir.
Thank you, ma'am, for your kind introduction. Am I clear, ma'am? And the slide is visible?
You're audible, sir. We're just waiting to see the slides.
This is visible, ma'am?
Yes, now it is visible. If you can just go to the slide play mode.
Slide play mode is problem, I think. If I go to that, it is not moving. I will just attempt that. It's viewable. It's visible, ma'am. Hello.
Yeah, I'm just waiting to see the slides. Yes, it is visible now.
It's scrolling, or can you see the second slides?
Yes, I could see the second slide.
Yeah, thank you very much for confirmation. A very good evening to one and all. Namaskaram. I'm extremely thankful to DBT for giving this opportunity, and also to the Ministry of Biosciences and the Director NOT for giving this opportunity to speak in this forum. I'm going to discuss on the marine natural compound and biotechnology perspectives. And everyone before my speaker, who spoke before me, had made my life simple and emphasized the importance of the marine resource. And if you see, the 60% of the marine animals is from 60% of the animals is from marine. When compared to the land animal of 5% or microorganisms of 50% or land plant of 12%, the whole large things components come from the marine. And in that, the coral reefs and a lot of other diversity systems are said to be having the huge biodiversity on this earth. When we move into a resource, everything right from marine bacteria to a large mangroves and resource, potential compounds has been identified a huge number of marine bacteria, which we will discuss in coming slides. But we have also identified huge number of fungus worldwide, and cyanobacteria has been utilized as one of the largest single cell proteins so far. Dinoflagellates have been explored for pigments and oils. Green algae similarly exposed for food and pigments, and brown algae for polysaccharides, and red algae for polysaccharides and carrageenan. And sponges also, sponges also been explored, and a number of compounds has been come out from the sponges, cnidarians, and bryozoans, and mollusk also, and anti-analgesic compounds has come from mollusk. Similarly, cancer medicines, drugs are patented from tunicate and echidorans and mangroves also. To make it concise, every phylum has a potential, and every phylum has a potential organisms for providing in marine natural products. So the classes of natural compounds coming from this marine compounds, you can see antibacterial, antifungal, antiviral, and anti-cancer, neurobiology, and anti-diabetic, and others. These are the major components coming from the marine systems, and the major components comes from the plant, from the marine plant, and the next goes to the microorganism, and the marine organisms. So let's explore. So this we will see why this difference is there. And the number of groups that has been categorized, the number of a pattern that has been made from this, the major compounds as identified from tunic sponges, which is about 52%, and the next dominant is in marine bacteria, and others are small and single digit for tunicate, marine algae, cnidarian, and echinoderms, as well as marine fungi and mollusks. So the major things are identified for sponges as well as for marine bacteria. And the category of compounds that has been identified so far is categorized into the large categories of is alkaloids, and the next category goes to the polyketides, and rest is terpenoids and proteins and unknown. And these are the major compounds identified so far from the marine system. And when coming to the number of compounds that is identified and proposed by the new country, it is growing very steeply from 2010s. The major country now, it is in China. They have come up with huge number of compounds from marine, and we have to find, India has to find a significant place in this list. The compounds which has been already approved from the marine sources, the marine sources is not new. It is from 1969 onwards. There are potential compounds has been identified and commercialized. In 1969, the first anti-cancer cybacterin was discovered and it was commercialized, followed by 1976 an antiviral compound by Virabin, and as well as from 2004 cardiovascular oil, support oils, omega-3 fatty acids from fish has been commercialized, and similarly the zinconetide from an analgesic compound has been identified from Cornus, which is commercially available. And in 2007, trabactidine and eribulin in 2010 and 2011. The last one is pelliptidipsin. It is approved in Australia very recently. So there are very less number of compounds coming, but there are very stable and standard compounds coming from the marine, and which has huge commercial applications and medical applications. So part of this, what the Marine Biotechnology Group in NIOT does is, we have a potential couple of ocean going fleets, and we participate in regular cruises. We do work on two to three groups of organisms. One is on the marine planktons, that's phytoplankton. And we work on the marine bacteria, including most particular to the deep sea bacteria. And for this purpose, we have developed an high pressure sampling systems and high pressure fermentation systems to culture the true barophiles and facultative barophiles. And this, under this project, we have developed a high pressure culture system rating of 350 bar, which can mimic 350 bar pressure. And we also developed a culture system and microbial sampling systems, which is 600 bar pressure rated, which can take samples from 6,000 meter without change in the pressure. And we have identified number of species like Streptomyces, Bacillus, Nigrospora, Astrotica, which are barotolerant initially, and we have cultured in varying pressure from 10 to 100 bar pressure. We have analyzed what we have done with all the species is we do a parallel analysis of cultivable as well as non-cultivable. By doing a metagenome, we do a non-cultivable analysis and do an cultivable analysis barophiles by the high pressure fermenting system. And so far, we have identified potential organisms, 348 number of barotolerant organisms, 180 species from bacteria, and 45 isolates from fungi, and 123 isolates from macronomyces. Out of this group of organisms, we have identified potential group of organisms like Streptomyces, which produce multiple proteogynin and triple proteogynins. Similarly, Nigrospira, which yields glycofulvin and spirobenzofuran. Streptomyces species, which give pyridazine and peptidazole molecules, and Astrotica species of sequestrin derivatives, and Notocordoxis for L-asparaginase, which is a glycine-free L-asparaginase, and a Bacillus forms which give lipopeptidic surfactant, which is used for industrial applications, and Streptomyces phenogrensis for cyclic peptide, Bacillus subtilis for antimicrobial peptide, and Bacillus classy for ectoin enzymes. And out of these things, we have optimized extracellular novel glutamase-free L-asparaginase from active bacteria, and this particular molecule has been identified from a marine bacteria, and those bacteria, the complete set of genome has been excised and cloned in an expression vector by recombinant technology, and that is ready for transfer for production of glutamase-free L-asparaginase, which is potential in cancer treatment drug. The second is a biosurfactant gene, which is used for industrial emulsification, which is extracted from a B. licheniformis. Similarly, the full genus is heterologously expressed in the E. coli production system, and the production was increased to threefold, and that strain is also ready, and that the whole clone is ready for the transfer. Similarly, in ectoin osmolite from a deep sea bacteria, as in Bacillus classy. We extracted all the A, B, C, and the 3 genes for ectoine production, and that has been also heterologously expressed in a bacteria, and the clone is ready for transfer. And similarly, we have also developed a multiplex PCR kit for real-time detection of major viral gene from the marine bacteria, and that has been transferred, and entrepreneurs working on the development on large scale. And also, we have worked on the development of deep sea novel microbes, which is purely living on hydrocarbons, and which can degrade or utilize a huge number of hydrocarbons as a basic source of food. And as a part of this, we have done also good wonderful science in this. We are culturing a barophiles and a barotolerant. An interesting barotolerant microbes and Bacillus subtilis we cultured in a high pressure fermentation system, and we analyze their morphology there and the molecular biology of this organism in different pressure. As we see, we have given 3 slides which has in different pressure rates, and 1 bar pressure and 50 bar pressure and 100 bar pressure. At 100 bar pressure, the microbes, the length, it goes on increasing. That means the division factor, the construction factor is not on, and it also produces terminal spores in the high pressure. And when you look inside the bactericidal activity, the total bactericidal activity changes to bacteriostatic activities in high pressure. So a whole genome profile expression system is changing, and there are new findings in the protein expressions in high pressure as well as. And this is all the adaptations of the microbes during the high pressure conditions. And this is the fatty acid difference during different pressure bar. I don't want to go inside detail. There are significant difference expressions in the fatty acids during the high pressure expressions. And the spectrum also of the subtilis during a different pressure express different expression patterns, but in the same spectrum. And similarly, we're also working on a marine microalgae. One of the microalgae is working on spirulina marine. We have a couple of species of marine identified marine species of spirulina, and we have developed a complete mass culture systems for a production of a single cell protein as well as in fibrocyanin, which is contemporary to the freshwater spirulina. We are also working on chlorophycine. Chlorophycine also one of the very good group of protein and lipid production, and we have working on Guinea salina, and we have developed a complete system for scaling up in mass culture for tubular photobioreactor, bubble column, and open raceway systems. And we have optimized the culture systems for this thing, and we have developed a technique for process for production of lutein from this. And we have a couple of patents out of this. One is European patent on the process for production of lutein, and a process for production of pharmacoactive nutrients. This all sequential expressions, and polyunsaturated fatty acids, which have a potential anti-TB activity against microbacterium, also being expressed. And microbial harvesting systems for concentration and harvesting of microalgae. The systems also all the patents from Germany. And these are the lutein that we identified from this potential strain. We have 3 kinds of luteins, all trans lutein, and a Z-glucin. Z-gluten was an isomer of that, and another one isomer is Z9, 9Z-lutein, with the different molecular masses. And because of this natural isomer is isomerization, they have a potential activity, and the major is in all trans lutein, and this technology also transferred to an entrepreneur. Similarly, we are working as a part of the huge group. We worked on the deep sea drugs from the sea program, wherein we collected a huge number of organisms from Andaman region, but that was an area identified for us to work. We collected. We have a good team of divers. We have qualified people to go around and collect samples. We have collected a huge number of samples, which is not from Schedule 1 species, and we have screened this samples for various activity and shared this activity with CDR and other participating institutes. More than 250 specimens belonging to various stacks have collected from AN Island by diving. And biological assay and screen results yielded that 26 antibacterial, 8 immunomodulator, and 3 antifungal, and 3 antitubercular, and 1 antihyperlemic compounds were identified, and those compounds are with CDRA, and they are looking into the further prospective of synthetic aspects. So when coming to the drugs from the macroorganisms, the first drug coming from the macroorganisms was 1969, and later it was the recent one is from from Acetian. These are the typical example for the problems and biotechnology requirements for the biomolecules. This actin is coming from Acidian, and which is a very potent molecule for anti-cancer treatment, and it is in the European market. Recently, it has been launched, and it is a commercial application for drug. And this, after identification of this actin from the Acidian, there was a huge scope, huge requirement for culture of this Acidian in the sea in open conditions. And the problem is the yield per annum, yield per year of the different seasonal variabilities, and the yield was very less when compared to the biomass, and it was very difficult to process the samples from the natural source constantly. At the same time, there was also synthetic part which involves 40 steps of synthesis, and which yield almost the same compound, but this was found to be very cumbersome and economically not viable to complete this process. What does the marine biotechnology role holds? As the technological advance, the Acidian was screened for the bacterial community for the production of same molecule. And it was interestingly found that as a bacterial symbiont found in this was producing a similar kind of compound. So there was an opportunity to synthesize, to culture this whole bacteria on the fermentation scale technique and produce the same molecule. And now the molecule is coming from a fermenter instead of coming from the bacteria. This is where the biotechnology makes it. Further, the same bacteria, similarly, this is another one from Alpidine coming from the bacteria, which is earlier identified from the whole source, and so now it is coming from the bacteria. And these are all the important compounds that has been identified from the organisms earlier, like tunicates, strobactin, dolastin, and all these compounds are coming from the source of marine organisms. But later, all these organisms were screened, and their bacterial, the symbiotic bacteria which is living inside this organism was identified, and now the production has been organized from this bacteria. And still the technological advancement is going on. The genome of this bacteria has been explored, and the specific genes involved for this production of this, if it was not a huge gene, and polygenic expressions, a simple set of genes are excised and are recombinantly produced. And now most of these things are produced from the bacteria. That means the original samples, the organisms they have collected the organism they collected the bacteria. From the bacteria they have imported the gene, and the gene now is expressed and put in a written expression system, and that expression system on E. coli is acting as a source of production. So that is how the natural product and the biotechnology interventions is working on the natural product to cope up the demand and to meet the requirement. And these are the new compounds from 2004 to 14, which 4 to 11, which is there. There's Elan chronic pain from Cornus, and Pharma cancer, that is Yondalis from E. turbinata, and similarly the breast cancer ESC from Alicornin, and the Seattle from Australia. This is all these companies are coming from one or two major players from the marine. And what does the future hold? The future hold in marine biotechnology, it search like a needles. It search continues. It's like a needle in a haystack because of the huge sample collections and the huge number of samples screened and the time frame to commercialize the unknown drugs. And in spite of this, doing this so much time, we have the still biopiracy open, and still we have to go a long way to get commercialization. But still, there are huge companies by the recent biotechnology interventions could able to make these things commercially viable and cut short these procedures. And we are producing number of compounds from the marine system, from maybe from the knowledge taken from the marine system, or the resource, that's the genetic resource, or the genetic knowledge taken from the marine system. That's it. And thank you very much for the opportunity. Thank you. I hope we have done in the time.
Thank you very much, Dr. Dharani. That was really nice hearing about all the biotechnological interventions that are there. We are aware biotechnology can do wonders if we start exploring, and marine life is a plethora of compounds which I think we have not even started exploring. And there is a great potential for us to tap this source so that we can explore new compounds and really look forward to, helping mankind in developing new type of compounds. So thank you very much. As mentioned,
Dr. Shilpi Gupta, thank you very much for your kind words.
Thank you, sir. Moving forward, as our previous speakers have reiterated, marine life is a mystery. There are a whole lot of compounds that are there which we need to establish, which we need to look for. And we're asking our next speaker to give his talk on actually deep sea and distant water living resources. What are the potential and perspectives? Maybe it will help us to finding a solution to the mystery. Dr. Hashim from Center for Marine Living Resources Ecology, I would request you to please deliver your talk. Thank you very much.
Yeah, thank you. I hope actually my presentation is visible. Screen is visible for you?
Yes, it is visible.
It's on full screen mode right now. It's okay?
Yes, yes, please go ahead.
Okay, thank you. Very good evening. Thank you for giving this opportunity. I would like to talk about the deep sea and water living resources and what is the potential and prospect. So since it is deep sea, actually what basically many people was not much aware about what is deep sea and what is deep sea fishes and how it's and what is ecology. So I'm just going to take some few minutes to explain all these points. So deep sea is can be one of the largest ecosystem on Earth, and which has up to 200 meters, that is called as a sunlight zone, that is epipelagic zone, beyond which it's considered as a deep sea. So deep sea has got many gradations, that is mesopelagic, bathypelagic, abyssopelagic, and adult zone. And the animals which lives in such an extreme environment with there is no light and high pressure has got a different kind of potential, which we can utilize those potentials. So these are just an introduction about deep sea and deep sea fishes. Coming to the deep sea fishes, which is my expert area, and you can see on the corner of the slide there are some pictures of deep sea fishes which look a little scary, but they are highly potential. And according to the FAO, actually, these fishes, which animals which is living more than 200 meters depth, which is considered as the deep sea fishes. And in 1976, actually, after declaration of Indian EEZ, the institute of in the country which was started giving the responsibility to explore these resources, on which actually the CMLR was a nodal agency for the explore these resources. Apart from some few deep sea shrimps and few deep sea sharks, all other deep sea resources are underexploited, exploited or unexploited. Beyond which, there are deep sea resources, deep sea fishes, mesopelagic fishes, oceanic squids, and deep sea sharks, which have a high potential for the exploitation. And we got actually how could we do this? This works actually. We have a state of art vessel called Sagar Sambada, which is the only vessel could do deep sea trawling in this country. She has commissioned in 1984 and built in Denmark, and she has completed her 399 cruises across the country and even beyond the national jurisdiction. And this is just a map is showing her coverage during last 3 decades. And these are some of the equipments which we use for this exploration, of which actually I would like to show you that deep sea trawl net, which can trawl even up to 1,400 meters deep, and chain back dredge for the exploration. I'm just going a bit fast because I got many slides. And coming to the deep sea fishes again, actually we have blessed with very high diversity of deep sea fishes in our waters. According to a study, we have identified more than 200 species of deep sea fishes, of which many are new to the science and new to the area. And we have identified many areas in Indian EEZ which is high potential in terms of diversity as well as in terms of its abundance. So you can see many species which there are promising for the commercial exploitation, of course, based on their volume. But whereas in the coming slide, I'll just tell you why we could not give just a green signal for this exploitation. Based on our study, just see there are areas which is high in potential of many species like Splisionica, Solnocera, and of course these are shrimps, and there are many deep sea species. Even in Arabian Sea, Andaman and Bay of Bengal waters, we have many areas which we have identified their high potential. But before going to the exploitation level, now during from 1992 onwards till last plan period, we were concentrating on to study the diversity and distribution and its abundance. And before we venture into a commercial exploitation level, actually we should know the ecology, how these species are existing in this kind of environment. Based on which, during this plan period, we started giving more importance to study their ecological level studies. Actually, apart from the normal, that is up to 200 meters, actually the area is completely different from the normal. Actually, it's not based on the the food chain is not based on the photosynthesis, and it's completely different, and in a microbial loop is involved, and many different others. So we should know how these species are existing in this area. So based on that, we started ecomorphological studies, which we have taken some of deep sea fishes and how they are partitioning their niche, how they are partitioning their food, and how they are existing in this area, which we could initiate those studies, and which has resulted we got a very good result, and same has been published in the high impact journal like Deep Sea Research and Sandia Marina. These are based two studies. One is on angler fishes, and other one is on the other deep sea fishes. And apart from the morphological study, we also venture into the otolith based studies. Actually, through the otoliths, we consider as it's basically it's a black box of a fish. Actually, we do have many balancing organs like Malleus intestine is like this. Actually, the fishes also got the otolith. Through this, actually, we could be able to differentiate the different kind of stocks of the deep sea fishes, even if it is from same species, which we got from different area, whether stocks are different or same, and how they are distributed. All these, how deep these species can dive. All these points we could be able to study. So this is just beginning of this study has just began. And apart from this, actually, the country has blessed with very good resource of marine mammals. Of course, we are not going to exploit these resources. We, based on our study itself, actually more than 18 species of mammals has been spotted throughout the Indonesian, of which actually there are many pockets which we can see high abundance, and their spottings are very high. So mammal-based tourism can be promoted if we the study could deliver. Actually, if we could able to find out a particular area which we can give for the marine mammal-based tourism. And these are some of the snapshots of during our study. You can see how beautiful these guys are, and it's really promising for the marine mammal tourism. And coming to the of course, we also have two big groups of islands. That one is Lakshadweep and other one is Andaman, of which the last plan actually we could study the biodiversity of Lakshadweep Islands, which has resulted more than 166 different phyla, different groups of beautiful marine world, which is a promising area for the island-based tourism. We have conducted more than 35 scuba diving in this area, and we have recorded many organisms, of course, apart from which actually many new species and new records we have made. And we also studied how the tides and temperature which is varying. And before we are venturing into this tourism-based business, we should know how much the impact of the changing climate is impacting this area. That is also one of the important points. So, of which actually we have taken the studies of how the water mass is changing, and And now you can see actually some of the very good, beautiful photographs which we have taken from the Lakshadweep island. And these are some of the publications which is we have come out from our study. And apart from the tourism, actually the area is highly potential for the marine aquaculture. So coastal, the marine ornamental fishes is highly abundant in this area. And based on our study, actually we have calculated how profitable to do a marine ornamental fish hatchery, and how profitable this, and which can be even can be a very good alternative income source of the islanders. So this is one of the promising area for the Bioeconomy to do the marine ornamental fish hatchery in Lakshadweep waters. And in collaboration with the CM of RA, actually in the last plan period, we have conducted a study in Andaman waters for the culture of black-lip pearl. Actually, you know the pearls, but normally it's the black-lip pearl is from a species called Pinctada margarifera. Actually, we could, these are some of the method I'm just skipping out, and we could able to the technology for the breeding the black-lip pearl we could do in Andaman waters, and which could be a income source for the islanders, which if it has established in the commercial level. And these are some, this was a site where we did our black-lip pearl culture in open sea. And some training program, of course, we have conducted for the islanders, and for how best they can involve in these programs, and how best they can do the earn the as an alternative income source. And of which actually there are one more resources which is called from the deep sea itself, that is myctophid from Arabian Sea. It is considered as more than 100 million tons of myctophis species, especially from a single species called Bendosima striatum, which has got a lifespan of less than 1 year, which is highly abundant in Arabian Sea alone, and which can be used as a ingredient for fish meal, and many products was developed based on our study. And it's of course it is not suitable for a direct consumption because of its high wax content, and it has high in PUFA and DHA, EPA, and there are many minerals. It's in the muscles, and we have developed many products like myctophid fish meal, myctophid oils, myctophid proteohydrazine, myctophid momos and rolls, battered and breaded products. Many products was developed during the last plan period from the particular program. And apart from this distant water resources, actually we have signatory to CCMLR, that is Commission for the Conservation of Antarctic Marine Living Resources. And based on their surveys, actually approximately 380 million tons of Antarctic krill, that is Euphausia superba, which is in the Antarctic waters, and which is a major source for the Antarctic food web, of which actually there are we have assessed the total allowable catch, and even India also can be the part of that one. And we got a 5.6 million tons of total allowable catch of Antarctic krills, which can make many products like krill oils and krill battered products, krill breaded products. Many products can be, but right now we are not utilizing these resources. Actually, we are only one study was conducted during 1995. That is our first expedition to the Antarctica. It was yielded with 565 kgs of krill on a single expedition, and we have made many products like krill cutlet, dried krill products, krill chutney, the whole dried krill. Actually, all these products was sold in our local market, and it was found to be highly profitable if we could do it in a commercial manner. Thank you. I was a bit fast, so I thought actually in 10 minutes I could not be able to cover all these slides, so I just hurried up. So please let me know if any kind of explanation is needed for any points which we have discussed. Thank you.
Thank you, Dr. Hashim.
Thank you.
That was now to give an idea about the fishery advisory services for blue economy, I would like to request Dr. Uday Bhaskar from Indian National Center for Ocean Information Services to kindly deliver his talk. Hashim, can you please stop sharing the screen?
Yeah, sure, please. I'm just...
Yeah, good evening. Able to see my screen?
Yeah, it's came.
Yes, Dr. Uday.
Okay. Yeah, good evening. Myself, Uday Bhaskar from Indian National Center for Ocean Information Services. And I'll be talking about one specific service which has got a great potential to be considered in the blue economy, that is Marine Fishery Advisory Services. This is the common minimum program with which Inquires has started way back in 1999. And then we went on to add many other services into our system. But today I'll be concentrating more on MFAS, as Marine Fishery Advisory Services. So just to state the mission and vision, because this is pretty important. Based on this, all our service activities have been concentrating upon. So we have a mission to provide data, ocean information and advisory services to the society, industry, government and scientific community through sustained ocean observations, constant improvement through systematic and focused research, information management and ocean modeling. So with this mission, actually, we started to serve various end users, mainly in ecosystem-related services, particularly PFZ, coral reefs, and harmful algal blooms, and ocean state forecasting services, disaster-related services, coastal geospatial services, data-related service and capacity-building activities. So with this, we try to serve the users which are shown on the right side, the fishing community, in the coastal states, IMD, Navy, Coast Guards, and ports and harbors, and then also research institutions, academia. So for this, we have a lot of data being archived at our center, and also we have got people running a lot of models, trying to generate various outputs which are being indirectly used for serving these end users. So as I told in the beginning, I'll be concentrating mainly on the fishing service as a part of this Blue Economy initiative. So this is pretty well known. I just put it, just wanted to say that this simple thing that we learned in our childhood has become the main source for this service, the so-called potential fishing zone advisories. So we all know that like the primary producers are being dependent upon by the phytoplankton, and phytoplankton are eaten upon by the primary consumers, and this becomes food for the secondary consumers. So this, the so-called simple food chain, is the one which we use to try to identify the areas where there is an abundant fish. So speakers prior to me has already spoken many things about the organisms that are available in the ocean and things like that, but I'm directly going to talk mainly about the fish service that we use and give to the end users. So this is the flow diagram which shows how exactly we are trying to serve to the end user. So how do we do the fish finding based on the remote sensing approach? So what we do is like we use ocean color monitor and then MODIS data sets mainly for getting the chlorophyll information. And then we also use sea surface temperature from NOVA series and then METOP. With these two, what we do is like we use the ocean color and SST mainly for identifying the regions of upwelling, and also which in turn tells about the regions where a lot of nutrients are available. And the ocean color is mainly used to find out the phytoplankton. So these two together, when you put together, we know that like this becomes a source of food and which are eaten up by the fish. So the fish tends to aggregate. So this simple process of using the remote sensing is used by INCOIS, and we generate the so-called PFZ maps, that is potential fishing zone maps, which we will be giving to the end users. So this slide shows the entire process that we do. So we depend on sea surface temperature. We can see the high resolution AVHR data that we get around 1.1 kilometers resolution, and then the chlorophyll from OCM and then MODIS as high resolution as 350 meters. Both will be using and then trying to identify regions like meanders, eddies, and then zones where we can clearly identify the upwelling. So these two regions, these two pictures from the satellite are being used, and then we have a set of people who are trained to draw those PFZ maps along these lines because these are the most productive regions. So depending upon the region from the coast, we try to give mostly from the coast, 25 kilometers away from the coast, so that the fishermen with different types of boats can also be accommodated with this particular service. So on the right side, you can see maps where it shows like we have drawn a PFZ lines, which is clearly identified from generally mainly from the fish landing centers or from the nearest lighthouse. So these are the indications that we use and try to provide these type of lines. On the top of it, we also try to generate maps of the currents, because we know the fish tends to move with the currents depending upon the time of the day. Like in the morning, it tries to go against the currents, in the night with the currents and things like that. So these two informations we try to give, and the bottom is the place where we can see it's a plain text. We provide this to the fisherman saying from so and so lighthouse at so and so direction at so and so depth, you try to if you go there, you can get abundant fish. So this simple information translated from the lab to the land is making waves in terms of having a huge economical potential. And the good thing is, like we try to serve this center information in different local languages, so that people need not struggle trying to convert things or trying to convert the information into their local language and trying to understand and then break their head. So that simple conversion is being done. Also, we tend to convert the units into the local followed units. For example, bara and things like that. So for the local language people, instead of trying to give in meters, kilometers and things like that, so that they're most familiar, and then they can directly go there and then start fishing and then get fish catch. So as I told, the advisories are given in multilingual in local languages and also in local units. So you can see the different sectors that we serve all along the coast, giving in their local language. We cover Malayalam, Kannada, Marathi, Gujarati, Hindi, Telugu, Tamil, and then Oriya. So all these courses are covered, converted into the local language and then sent to the people each day immediately after the processing. The processing starts sometimes 12 o'clock in the afternoon. The moment we get the pass in our ground station in our center, immediately the pass is converted into the sea surface temperature and then chlorophyll within 10 minutes. Immediately the processing chain starts, and then the PFZ lines are drawn, and all these informations are derived and then immediately passed on to the local people. And then we tend to use all available sources for dissemination, because generating is one aspect, but dissemination and see to that it reaches to the fisherman well within the time is another aspect. So we use all ICT to reach our end users. We use different dissemination modes like SMS, telephones, mobiles, and then fax. And we also have something called digital display boards, which are kept at the coastal stations, where the fishermen can directly go and then see. We have even come up with mobile apps that is developed, which is fisher friendly, and they can happily get this information on their mobile apps, the WhatsApp, local radios, local TV channels, everything that is possible that we are trying to reach to the end user. And why I told this one particular service has got a huge potential for being the candidate for the Bioeconomy is like this one example. We have conducted lots of validation experiments to see how much of potential this particular service is having. So what we did is we conducted experiments by giving the PFZ notified areas and non-PFZ notified areas and asked people to go by different boats and then try doing fish catch. So as already done, like the PFZ notified areas has really got pretty good potential compared to those regions where there is no PFZ lines notified at all. So the right side table shows the economic benefit that the PFZ service is being, for the people that is being used for the service. So you can see there is a huge net profit for the people where they have gone to the net PFZ identified areas, something like 2,82,400 rupees compared to those people who have gone to non-PFZ notified areas, just yielding only 68,560. This has been done pretty many years back, but this still holds good even today because we continue to take feedback from the fishermen and then try to see how we can adopt and then improve our services so that their profit is pretty much staying as such and they're not dissected by going to the areas where we are trying to identify. So if we can see the summary of some of the validation results, the estimated success rate is something like 80 to 90%. And there is huge reduction in the search time. If it is not given, people have to go there, stay overnight, spend huge amount of time, and then they are not still sure whether they can really get good catch or not. So there is a reduction in the search time by 60 to 70%, and then increase in the fish catch by 2 to 4 times. And then the net profit is also increasing by 2 to 4 times. So something like 600 plus validation experiments have been done during various plan periods, and the results are quite amazing. So on an average, if you can see for mechanized boats, it's something like 6 lakhs income is there. For a motorized boats, 1.95 lakhs income is there. And then small motorized boats, 1.65 lakhs income is there. So considering 27% of the boat owners are using our PFZ advisories, there is a saving of around 163 crores itself only for the Kerala coast. One example I'm just taking here. So if 100% of the mechanized and motorized boats are operating based on of PFZ fisheries advisories, there's something like saving of 600 crores on the diesel, and then also the valuable human efforts savings are there. And extrapolating this scenario to the national scenario, it indicates a savings of around 1351 crores for 27% usage and 5,000 crores for 100% usage. So if a simple satellite launch, assuming 4 years of life, it almost comes to around a huge benefit of 5,000 crores per annum for this single service alone. So if you can see the impact of the advisory with and without this one simple flow diagram which can show. So without the PFZ advisories is quite less cash and profit and higher search time, more fuel consumption and increased carbon emission, and then huge human efforts are there. So compared to that, if you use the PFZ advisories, so 2 to 4 times higher catch and profit is there, and reduced range of carbon from 3.5 to tons to 0.06 tons to 3.45 tons, and savings are ranging almost from 2.47 liters to 1293.53 liters in terms of diesel. So there's a simple, clear demarcation of the benefits, and this is recognized by the international community as one of the matured operational application of remote sensing data and providing the service from lab to land for a common man. So, and also estimating the benefits as such by different agencies. We have involved various agencies asking them to do an independent survey, because we being an agency giving this, we can talk saying it is really helping the end user, but we want independent agencies also to do this analysis for us. So different agencies have come up with estimates of how much environmental benefits this particular program is having. For example, NEIP has estimated that there is a cutting down in the carbon emission by 150,000 kilos. And then validation experiments have shown like there is huge savings in the diesel. And NCR report has come up with an estimate that this is annually there is a savings of 36,200 crores, coming to around 0.5% of GDP. That's a huge economic benefit from this particular program, because of which the government of India has identified this as a common minimum program and asked us to continue free of cost to all the fishermen in the country. And coming to that is the environmental benefits. This coming to the economic benefits of the PFZ, and then ocean state forecast. Because earlier we used to give PFZ alone as a service, but the safety of the fishermen is also of very importance to us. So we have another service called ocean state forecast that we have included along with this PFZ, so that along with the economics, they are also safe. So since I'm just only covering the PFZ part of it, I'm just going to talk the overall economic benefits in all the industry with respect to the PFZ is roughly estimated around 3.5, 3.7 trillion. And then annual economic benefit coming to 34,000 to 50,000 crores. And then additional benefits of fishermen coming to around 3,000 crores annually with the investment of simple 32 crores. So per boat also you can see like huge success rate is there, almost 80% success rate, profits are 3 to 4 times, and such time has been reduced by 30 to 70%. So this entire thing has been surveyed and then recorded in an NCEAR report, which is available on website for people to download. So this really is a contender for to be a part of blue economy. But the care need to be taken that people should not be over exploiting the fisheries in our coastal areas. Accordingly, we also follow the norms given by the government of India, asking us to try to provide the ban periods. So accordingly, during the monsoon, pre-monsoon, May to June, for east and west coast, different ban periods are defined. We maintain these ban periods and we don't give any advisories to fishermen. And we also tell them not to go during this ban period, because juveniles and all will be there which are got caught, and then obviously this might result in huge loss once there is no matured fish or exploitation. So provided if you follow all these norms and try to do matured way of fishing, and also try to exploit this in the open ocean, because right now we are only covering it in the coastal. We also have plans to extend to the coastal, beyond the coastal regions. Then the economic benefits are going to be even more than what is estimated over here. And as I told in the beginning, we try to get huge amount of feedbacks from the fishermen and try to incorporate and then see how we can reach more. And then in case we are having any issues with our forecast, we try to rectify them and things like that. So these are type of feedbacks. I just put it there. Thank you. If any questions are there, I can just answer, because I just hurried through owing to the paucity of time.
Thank you, Dr. Uday Bhaskar, for such wonderful talk. Now I request Dr. N.V. Vinit Kumar, that from the
Hello. Can you hear?
Yes, yes, we can hear.
Yeah. Can you see my slides?
yes, we well, please do in full mode, presentation mode.
Okay. First of all, I would like to thank DBT for organizing this Global Bio India 2021, and also I would like to thank the organizers and my own Ministry of Earth Sciences and our director NAOT for giving me this opportunity to talk in this forum. So today I'm going to talk about this open sea cage culture of fishes and seaweed culture technologies, prospects and that opportunities in India. if you before giving any other introduction about our coastal wealth, all these things, let me directly go into the things that we are working in Andaman Nicobar Islands. We have a center called Atal Center for Ocean Science and Technology for Islands, which is under the National Institute of Ocean Technology of Ministry of Earth Sciences. So we are working on various aspects like open sea cage culture, marine microalgal algal culture, microbial culture, marine microalgae culture, then pollution monitoring, as well as environmental impact assessment, like that. Wherein today my talk is only on this open sea cage culture and seaweed culture, which is being done in along with my colleagues at NIOT Chennai in Tamil Nadu and in many other states, as well as in Andaman, especially in Andaman Nicobar Islands too. So if you see our global fisheries and aquaculture status, as per the recent 2020 status, state 6, we can see that it is the fish production reached mostly of about 179 million tons in 2018, which is of about $401 billion US dollars, of which about 82 million tons, which is valued about 250 billion, came from aquaculture, which is about 46% of total production. If you see in that 179 million tons, 156 million tons were for human consumption. Rest of the amount is used for various other processes like non-food uses like fish meal and fish oil. Similarly, if you see the right side our that map, from the 80s onwards, that capture fisheries, that orange colored one, it has leveled off, and the presently the aquaculture is meeting the requirement of the people. So in that, inland aquaculture constitute dark blue color, that is showing constituting more, and mariculture, which is also constituting, but in lesser than that of our inland aquaculture. So we have a very big coastal line, and there is a lot of potential for our mariculture opportunities. So it will help us to develop various technology for the people, particularly to meet our protein requirement. Present per capita consumption of our fish is about 20 kg throughout the that is when compared to the world, but India is still not meeting this 20.5 kg per year. So now the major producer of global fish production is by China, but excluding China, 34% is contributed by Asian region. So if you see the Indian marine fisheries potential, our estimated potential is about 5.3 million tons, but our present production is only 4.17 million tons. That is, we are almost achieved 78%, 80% of our potential we are already achieved. So, so we have to meet our demand of protein fish from the aquaculture aquaculture point of view. That means we have to develop a lot of culture technologies in the open ocean conditions. So that will help us to develop our technologies and transfer the technologies to the people, so that we can produce a lot of food, that fish food. So inland also, if you see that 17 million tons we are producing, whereas which is only 56% of potential. But there is a lot of inland areas which is not yet been used for production. Similarly, India's fish production is only share is only about 7.5%, 5% to the global production. That is contributing about 1.24% of India's gross value added products. So, which is almost a considerable amount in the our gross GDP. Similarly, if you see this 156 million tons from the 18th, that if you see that per capita consumption of fish is only about 11.5 kg. Presently, it is about in the year 2020, it is about 20.5 kg. So, in the coming years, during the 2030, suppose there will be a lot of requirement of fish protein, which can be met from the aquaculture based technologies only. So this is what this graphs also shows that present that the population is about nearing about 7.8 billion, which by the end of 2030 it will become around 8.25. So there will be a difference of 25 million to 27 million tons of fish required, which can be met only from the aquaculture based technologies only. So in this background, our NIOT is working on various aspects of this mariculture aspects. So particularly from the year to 2000 onwards, we are working on different technologies. First one is we initiated our work with this fattening of farming up and fattening of spiny lobsters. Then similar time, we have worked on the mud crab fattening technologies in Andaman Nicobar Islands in cages. Similarly, we have deployed fish aggregating devices in Andaman Nicobar Islands, and we also worked on the various design and construction of and the emplacement of artificial reef, which we have deployed in Tamil Nadu coast as well as Orissa coast. And we have developed these open sea cage culture technologies, and we have done so many works in the coastal areas of Tamil Nadu and Andaman Nicobar Islands as well as Andhra Pradesh coastal areas. We have deployed many cages in different places, which is of about 9-meter dia cages. These are all indigenously. Now we can purchase these cages in India itself, and we can deploy in suitable locations in the coastal areas. And there are various species of fishes being cultured in this, particularly this fish cages. The cobia, the Rachicentran canadam, and sea bass, which are giving best results in these open sea cages. These are all the designs you can see, which is developed, which these can be purchased from the local Indian industries itself. So where previously it was imported, later we have indigenized all these parts, and now we can even deploy in deep sea conditions also. We have deployed the cages in different places like Kottachatram, Olekuda, North Bay in Andaman Nicobar Islands, and Tiplipalam in Andhra Pradesh. Olekuda is the semi-enclosed area. This is North Bay is particularly fully protected area. That the Tiplipalam and Kottachatram, it is open sea environment. All these areas, wherever we deployed these cages, open sea cages, they survived for very long years and produced good results. And now we have developed technologies for even larval, small, that fingerling rearing within the 9-meter dia cages by developing 2-meter dia cages, so that this nursery cages, in the nursery cages, small 0.2 gram, that is 2.5 centimeter fishes can be seedling can be introduced. Once they grow up to 10 centimeters, then they will be directly introduced into the bigger cages. Thereby, you can we can culture the fishes in the same system without having separately having that nursery rearing facilities in the hatchery itself. So once we are getting the seeds, we can directly introduce into this small type of small 2-meter that cages, so that we can grow up to a harvestable size. So for this purpose, we have collected seeds from SIBA and CSMCRI and RGCA and many places, and deployed these cages in different places where we introduced these fishes, and we have done a lot of cultures in different places during the different years. So if you see this sea bass culture, we have demonstrated in Andaman Nicobar Islands. We could able to produce around a ton of fish, and we can involve the local beneficiaries, and they actually very much interested, and we train those people also. And imagine this fishes were grown from the size of 0.22 gram, less than 1 gram size, to around 1.2 around average size of 700 gram. Some fishes were about 1.2 kg also within the period of around 11 months. Whereas you see in the harvest of Kottachatram in 2012, we have harvested about 1.5 ton of Asian sea bass. Similarly, in other place, in Walikuda, you can see the harvest of cobia, Rachichandran Kannadam. We got that farm gate price of about 325 rupees per kg in that harvest, so that it was about 6.5 ton total we harvested from the different cages. The various species are cultured, particularly milk-based pambanos and cobia fishes were cultured. Based on the fish quality or preference, it fetches different rates. The cobia is one of the successful fishes which can grow up to a 4 kg within a short period of 8 months, whereas the other fishes which will grow very slowly. So this cobia gives very high profit for the fisherman. Based on our experience, we have actually done the geospatial mapping, for where and all we can culture the fishes, that we can deploy the cages in Indian coast. In that, we found out most of the places which is highly suitable, and there are pockets of unsuitable and least suitable places. These are all defined based on our water quality criteria, as well as that environmental, that is wherever the depth, we have considered more than some 15 parameters. Apart from that, we have, based on this, we have developed so many cages, and also these are the fishes we have cultured: Latus calcarifer, that is Asian sea bass, cobia Rachichandran Kannadam, and Pampanotrachinotus beloki, and Chanas chonos milkfish, and parrotfish, Scarus gabon, and giant ravelli, that Caranx ignobilis. So this is one of the success story from our NIOT. Similarly, we have initiated our work initially with this culture of this lobsters in various places of Tamil Nadu, and even sometimes we have given a demonstration in other places also, other than Tamil Nadu also. And this also gave a good profit to the people. Same time, we have done the demonstration of mud crab fattening in Andaman Nicobar Islands And some places people were very much interested, and they could the self-help group could able to get around 10 lakh rupees within the period of that 6 months period in North Andaman areas. These are all the different cages, locally available material, bamboo cages as well as FRP cages we have designed for them, and this is the FRP cages deployed at Pulicat Lake. Next, coming to the seaweed culture, you know that seaweed culture, a lot of introduction has been given. In India, we have in world over around 10,300 species of seaweeds are reported. Among that, 221 are commercially important, and in that 145 species used for food, and 110 species are used for phycocolloids. In India, we have about 1,153 species. In that, 282 species we can found out from the Tamil Nadu coast as well as Gujarat coast. So our in the seaweed culture technologies, we are working on various aspects, particularly biodiversity estimation and stratigraph estimation, then finding possible sites for seaweed culture, then also development of various products from the seaweeds, particularly biostimulant, sodium alginate, and cosmetics, face pack, kajals, and similarly food product that is from edible seaweeds, and also biodegradable plastics also we have developed, as well as we are also working on extraction of these fatty acids from seaweeds also. So these seaweeds are used in different aspects. Previous speakers also spoken about it. It is used in biofood, biofuel, and biogas and biofertilizer production, and they are used in cosmetic, fertilizer, and food industries. And based on the importance, now if you see the global production of seaweed, it is estimated about 32.4 million tons in 2018, which is about 97% is coming from culture alone. Only 3% is people are collecting from the nature. So the China is actually a major producer of seaweed, and these are all the 5 countries: China, Japan, Indonesia, Philippines, and Malaysia. And Vietnam are the major producers of seaweed. The major seaweed species cultured are Euphemia, Kappa physis, Racellaria, Saccharina, Undaria, and Pyropia, and Sargassum. So if you see the that by region, Asia Pacific region, which is producing more seaweeds in the world, it is expected that in the year 2024, the red seaweed will dominate. Even now, the red seaweeds are produced in more quantity. By application wise, seaweeds are used for human consumption, but then hydrocolloid, fertilizer, animal feed, and others. Similarly, China, as I said, it is a major producer, and there are Korea also producing about 8,000 wet tons of seaweeds from 3 different seaweeds. Japanese produce more than 6 lakh tons, that which is in that mostly nori is the important product, which is you can see they are using it in all the food that is food packings and so many other things. So agar is the one. Agar is produced from seaweed. This agar is the seaweed is the only source of agar. So there is a requirement of seaweed that is agar production, which can be met from the seaweed culture in the open sea environments. So seaweed, as I said, which are abundantly available along the coast of Tamil Nadu, Gujarat, and you can find out from Mumbai, Ratnagiri, and so many other Villingham and Pulicat Lake in Tamil Nadu, as well as Chilka Lake in Orissa. In that is 153 species in India. 60 species are commercially important species. Mostly this Geldiella acerosa and Geldiella gracilaria directly collected from the sea, um, which are and sargassum and turbinate are also collected during the August to January in the southern coast. About 16,000 tons of sargassum and turbinate has been reported from the Indian waters. So Indian industries, seaweed industries are mostly cottage industries. They are using that seaweeds for jelly, that agar production, particularly from Gelidella acerosa and Gracilaria adalis. And that other seaweeds, sargassum and terminaria, are used for phycocolloids. And so that the total production of seaweed in India in the year 2000 itself 6 lakhs ton. Later it was reduced. Now it is again increasing due to various culture strategies. So the first commercial that cultivation of seaweed in India is embarked by PepsiCo. Later now at Mandapam, Parkway side of Mandapam area in Ramnad district of Tamil Nadu. Whereas presently that Aqua Agri, one company is doing that promotion along with CSMCRI, Marine Algal Research Center at Mandapam. So they have initiated the seaweed cultivation with Euchima cottony and Hypnea musiformis in 100 hectare area, and they could able to produce around 100 tons per hectare. And there are so many industries based on their the zone that industries established for extraction of algae and so many other this one. This seaweed culture is one of the eco-friendly activity which absorbs treat the coastal pollution as well as which absorbs a lot of carbon dioxide. Based on that, we have done so many surveys in Andaman Nicobar Islands, and we could able to collect around 60 species of only alone from Andaman Nicobar Islands, south, particularly from the South Andaman. These are all some of the species we have recorded from Andaman Nicobar Islands. And these are the seaweed farming activities we are doing in Andaman Nicobar Islands. This is the first of its kind in Andaman Nicobar Islands. Previously, some studies they have done in Andaman Nicobar Islands. Mostly it failed, but recently we could able to identify particular sites, particularly Chidiatapu, and we have done the culture of Gracilaria adalis that is available in the local environments, so that they can be cultured without any effect to the environment, without introducing any other exotic species from the other mainland or other countries. We are using the locally available seaweeds to culture. Last month we have got very good production. Small a quantity of 2 kg is introduced into the small, that is this cages, 2 to 3 kg in 2 rafts, small rafts, bamboo based raft, and we could able to collect about harvest about 42 kg in within about 2 months time, 6 months time, sorry, 60 days time. So so there is a potential for culturing of seaweeds in Andaman Nicobar Islands. You can see further we are planning to culture the seaweeds in about 40 rafts. Further, during the season between that November to May, which is good season for the seaweed culture in Andaman Nicobar Islands. Other times, mostly that due to cyclonic weather condition and lot of rains, we could not able to culture these seaweeds in successfully in Andaman Nicobar Islands. These are all the activities we can find out. We could able to produce seedlings, and now we are developing for that seed bank for that particular area, so that we can take the this seaweed culture into next pilot scale culture, so that we can introduce around 200 wraps in that in those areas, so that we can give next level. We can give this technology for locally developed technology for that local people, and we can set up the small scale industries for processing various product. We are also similarly working on biostimulant. Recently, we have tested this biostimulant extracted from the sargassum along with this our gracilaria, and it produced good results in the that vegetable plants, and you could able to culture that it could able to give 30% more that is profit or fruit for the in okra plant also. Similarly, we are testing this one in the through other institutions also. Locally, Central Island Agriculture Research Institute is there. We have given our product to test it. They also found out these products developed from this by NAOT is giving good results. Soon we will get the certification from there. So based on that, Indian now these are the activities we are carrying out in terms of seaweed culture in Andaman Nicobar Islands. So government of India also actually under the Blue Revolution scheme, they have allotted around 3,000 crore for the year 2015.
Dr. Vineet Kumar, I'm really sorry for interrupting. If you could please finish up. Thank you very much.
So based on that, similarly PMMSOE also we could able to see that we want to develop, and it is our target to achieve 11,000 that is 1,70,000 tons by 2025. I think based on the technologies developed by ourselves as well as by the collaboration of various institutions, CSM, CRI, CMFRI, and SIBA, and NAOT, NAO, and we could able to achieve the targets which is set in the PMFSY program. With this, I am completing our presentation. Thank you very much. Thank you very much for madam for the opportunities.
Thank you, Dr. Vineet Kumar. It was really insights, you gave about the fish cage culture and the seaweed culture, and I'm sure it will help farmers in enhancing their produce. Also, seaweed is a great resource for harnessing important metabolites. So that was really insightful. Thank you very much. With this, we come to the close of our session. We had really enlightening talks today. We covered a range of subsectors from science and technology, biotechnology, remote monitoring, mysteries of deep sea seaweed and growth, and fish cage, and how the cultures and produce could be enhanced. I would really like to thank all our panel members, speakers, for sharing their work, giving us insight into the amazing world of life. I would also like to thank all our audience who have joined us for this session through the virtual mode. Thank you really all. And I apologize for the little overshooting in the time that we had. I think the talks were so enlightening that, there was no way we could have stopped in between. And thank you very much. And hopefully in the next edition of Global Bio India, we will be able to meet each other in person and not connecting through this virtual platform. Have a great evening and good night. And thank you to all.
Thank you. Thank you very much. Good night.
Thank you all.